All right. Thank you all for standing by. And I'm just making sure I'm recording. Yes, I am. Thank you all for standing by and welcome to our webinar entitled Discovering a Potential Treatment for Health Effects of Algal Toxin Exposure. This is a webinar series called Freshwater Science that will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today are Dr. David Kennedy and Dr. Stephen Haller, both associate professors in the Department of Medicine at the University of Toledo. Dr. Kennedy's lab focuses on how the heart, kidney, and liver regulate inflammation and other cell damage, especially over the course of chronic illnesses. His team is particularly interested in finding preventative and therapeutic approaches to these diseases, including strategies to address the cellular damage caused by harmful algal bloom toxins. Dr. Kennedy joined the faculty at UT in 2015 after working in a number of research positions with the Cleveland Clinic. Dr. Haller lab is particularly focused on kidney disease, how inflammation and fibrosis play into the progression of end-stage renal disease, and what therapeutic approaches could halt or delay this progression. Like Dr. Kennedy, he is also interested in these questions in the context of harmful algal bloom toxins, especially in patients with pre-existing health conditions. Dr. Haller joined the faculty at UT in 2015 after a postgraduate fellowship with the American Heart Association. We're delighted to have both of them here today to talk about their research. But before we do, just a few things about the webinar logistics. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20-ish or so, I'll conduct a question and answer session. If you have any questions, feel free to use that chat feature at the bottom of your screen, and I will collect and pose all your questions to Dr. Kennedy and Dr. Haller at the end of their presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. That always helps us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. David Kennedy and Dr. Stephen Haller from the University of Toledo, who will present Discovering a Potential Treatment for Health Effects of Algal Toxin Exposure. All right,
Super.
Dr. Kennedy, Dr. Haller, thank you so much.
Thank you very much, Jill. Can you hear us okay?
You sure can.
Awesome, great. Well, thank you, everybody. We saw the attendee list, and it's just great to have everybody. Lots of different states represented, different agencies, institutions, and so thanks everybody for spending a half hour with us today to talk about this. We love talking about it, and this is the 10-year anniversary for the water crisis in Toledo. I think that goes, sort of, I think if you're attending this webinar, you know everything about that, and I don't want to spend a lot of time on that, but it just gives us a, opportunity to kind of take stock of where things have been in the last few years and think about, some of the solutions that we want to focus on. And obviously, you don't need to spend too much time on this slide either if you're here, but this is just to visually demonstrate that harmful algal blooms are on the rise throughout the United States and all over the world, and so this is a very pertinent topic that we just don't want to sit here passively and wonder what to do, but want to try to create solutions to these issues. This is also a slide that we like to show. This was, I believe, it was about 2019 up in Monroe, Michigan, from our friends at Great Lakes Outreach. And this really gives us, again, a chance to sort of pause and to think about who's interacting with the lake. And, I highlight here on the slide, there's a sign on the beach warning, folks not to, be in or near the water. So I think that, we have some work to do and challenges You know, it's also unfortunate that, you know, people shouldn't have to, you know, drive 6 hours to, you know, to find clean water to recreate in. So anyhow, that again just underscores sort of the some of the issues that face people in terms of their exposure to harmful algal blooms and what we need to do about it. So when you think about how we're exposed, there's, you know, common routes of exposure. So we could think about ingestion, you know, through drinking water or contaminated food or water or even supplements. Our lab has been working a lot on inhalation exposure, breathing in aerosols generated from HABs. And then also, again, through a lot of this is through generous support from Ohio Department of Higher Education's Harmful Algal Bloom Research Initiative to look at dermal contact as well. So you know, these are the main routes of how things get in. And when you think about what do harmful algal blooms, what do the toxins affect? You know, there's been, you know, for some of the events that have occurred, have led, you know, maybe to the misnomer that these are just liver toxins. They're not. They affect every organ in the body. So they impact the lungs, they impact the liver, they definitely impact the GI system, also the brain, and nervous system, even in the heart and in the kidney. And we'll focus on some of these things throughout the course of this talk. But the key thing that I've, you know, highlighted here is inflammation as sort of a common theme in each of these organs and how HAB toxins impact that. The CDC is you know, helped lead a lot of efforts in terms of you know, what healthcare providers are you know, have access to and in terms of reporting. So I've linked this down here. You can see they have a physician reference guide for, you know, what to do with you know, exposures to harmful algal blooms. And this is tracked here through what's called an ICD-10 code. So healthcare providers can, you know, diagnose, or when they diagnose and record illnesses, they can do that through these codes, and that allows us to go into these databases and pull those codes out and kind of take a look and see, you know, who is getting exposed, and what's happening after they are exposed in terms of illness. And I'm going to just briefly go through some of the most recent data. This is from 2021. It was just published last year. This is the CDC's data. So in 2021, there was 368 HAB events. That resulted in 117 human illnesses and over nearly 3,000 animal illnesses in 2021. I know there's some attendees on the veterinary side of things, and that's definitely an important area. And if you look at this, so of these 368 HAB events, about 186 actually had toxin testing done. And microcystins were detected in about 166 of those, so nearly 90%. So microcystin is definitely one of the most predominant toxins, although you can see some of the other culprits there as well. And if you look at the people who are exposed, this is breaking those exposures down, again, the 2021 data by age group. And what you can see is that nearly half of the people who are exposed are under the age of 18. And then the other, you know, next largest group are those 18 to 45-year-olds at about 39%. And what do they complain about? What are the illnesses? Well, by and large, gastrointestinal is one of the largest. Generalized, you know, symptoms, dermatologic, it can include rashes, ear, nose, and throat, cough, cardiopulmonary, et cetera. So you can see that it's not one thing. It's complex, and it's really not very specific. And we want to talk a little bit about why that is. This is data from our group at the University of Toledo. We published, again, we talked about those, you know, patients under the age of 18. So this is a case series of patients that we've had at the University of Toledo. And again, if you look at their primary symptoms, they track very well with what the CDC reports. Some, generalized symptoms, fever, headache, but then also some dermatologic and then also respiratory symptoms. So what are the mechanisms that link these illnesses together? And what I'll say is, without getting too much into the weeds, microcystins in particular gain access to cells to what's called organic ion transporting proteins. So after they gain entry to cells, what's their target? And I've, schematically demonstrated this over here. Almost every single process that goes on in our bodies is regulated by what are called kinases, which typically turn things on, cellular processes on, and then phosphatases, which turn those signals off. And microcystins are a really potent inhibitor of this off signal. So I like to say, imagine, holding the breaker box in your house always in the on position, never being able to turn it off, and having things go a million miles an hour, 24/7. You can start to see why inflammation and oxidative stress are sort of the result of the disease. This gives us insight into that mechanism. And I put this here again just to kind of visually show. On the right here is this kaleidoscope. It's a pictorial presentation of all the pathways that protein phosphatases have. So these protein phosphatases, again, are the target of microcystins, which are, by and large, the number one toxin that's present in our legs, and they control thousands of important cellular processes. So all these processes, when microcystin inhibits that phosphatase, they're not able to turn off. So you get that's why it's not just a liver disease. It's or the liver is not the only organ affected, although it certainly is. There's a number of different organs that we highlighted, and Dr. Haller is going to talk a lot about the kidney too, because we think that's an important target to look at. So, we talk about tests and treatments, and, this is again from CDC, and this is what we think is a knowledge gap. So right now, if you look at the state of the science, they say medical care is supportive. There's no known antidotes, and there's no clinically available diagnostic tests. And we think this is an area where we need to move the needle. And today we're going to highlight one of those, areas that we think is a potential therapeutic for exposure to these diseases. So what are we doing to improve this? As a background, microcystin exposure alters the gut microbiota. All right, so those are the bugs that live commensally in our GI tract. And these alterations we know have significant impact on the development of inflammation, gastroenteritis, and hepatotoxicity, as well as some other downstream target effects. So in different liver disease models, treatment with probiotics in other areas has been shown to decrease steatosis, that is the liver holding on to fat. The liver is not meant to hold on to fat, but in different diseases it does, and that impairs its ability to function normally. And probiotics have improved things like insulin sensitivity and have been able to reduce hepatic inflammation. So we have a group of researchers here at the University of Toledo, Drs. Huntley, So, and Kang, who are in some of the other departments here at the University of Toledo, who have identified and isolated novel microcystin degrading bacteria from Lake Erie. So these were really very sort of novel and clever ways of looking at, seeing how the lake sort of heals itself from these toxins through the bugs that are out there, the bacteria. And that really led us to the question if these microcystin degraders could provide novel therapeutic strategies for exposure and organ damage, sort of like a probiotic. So that leads us to the overall hypothesis that we had, that microcystin degrading bacteria that we've isolated can be used as a potential therapeutic strategy to attenuate the adverse effects of microcystin exposure and organ injury. And now I'm going to turn things over to Dr. Haller. We're actually in the same room. We have different backgrounds, but I'll let Dr. Haller take over the presentation.
All right, thank you, everyone. Okay, so let's dig into the data. So now we're going to talk about determining the effect of microcystin degrading bacteria on microcystin toxicity in vitro using human liver cell line. So I would like to call out Dr. Apoorva Ladd. She's an amazing postdoctoral fellow in the lab, and she performed a lot of the studies I'm about to talk about here. So we used human liver cells, or Hep3b hepatocytes. They were exposed to the cyanotoxin degrading bacteria for 30 minutes, and then followed up with treatment with 10 micromolar of microcystin LR for 24 hours. And then we harvested the cells for analysis. So here what I'm showing you is that the microcystin degrading bacteria actually increases microcystin LR degradation in normal hepatocytes. So on the left hand here, we're looking at concentration in parts per billion. On the y-axis, x-axis are the different treatment groups. So in green, you've got microcystin, 10-to-1 bacteria, with microcystin LR 50-to-1 and 100-to-1. And you can see that the microcystin concentration is actually going down in the supernatant. And then interestingly, when you look at the actual cell pellet, you can see that the concentration of the tetrapeptide, so this is a breakdown product of microcystin, is actually increasing. And just to give you a better idea of that, after mass spectrometry analysis, so here you see microcystin, we were able to show, and this is working with the close collaborators, Dr. Julie Westerink at Wayne State. You can see here we're able to show the microcystin is actually broken down into that tetrapeptide, and that's what I just talked about, that increased in concentration in the cell pellet. So here, microcystin degrading bacteria protects against MCLR-induced hepatotoxicity in human liver cells. So here we're looking at OSMR, which is a potent marker of liver injury. And you can see that MCLR here in green, it goes up. And then when you look at all the different concentrations of microcystin with the degrading bacteria, it goes down significantly. And same thing for SERPINE1, another major liver injury molecule. It goes up after treatment with microcystin and then significantly downregulated when you follow that up in treatment with the bacteria. So here, microcystin degrading bacteria protects against MCLR-induced inflammation in human liver cells. So here, TGF beta, a prominent inflammatory marker. Again, you can see increases with microcystin. And then when you follow that up with microcystin and the degrading bacteria, you get a significant decrease. Similar story for TNF, another major inflammatory marker. You get the increase with microcystin and a significant decrease, particularly over here at 1 to 50 with the when you add in the microcystin degrading bacteria. So now we're going to switch gears a little bit and talk about determining the effects of microcystin degrading bacteria in vivo in our model system. So here again, I want to give credit to Dr. Apoorva Ladd for performing the majority of these studies. So we took bulb C mice, and they were 4 weeks on cyanotoxin degraders in drinking water, followed by oral gavage of 500 micrograms per kilogram of microcystin. We then harvested organs for analysis. So here, what you're looking at is that treatment with microcystin LR degrading bacteria actually improves toxin metabolism. And this is after mass spectrometric analysis in the urine. So you can see here on the left, microcystin and then microcystin cysteine, the non-toxic variant. And then over here with the probiotic, what you see is that when you follow that up with probiotic treatment, the non-toxic variant actually increases the microcystin cysteine. So it's trending to increase here in that non-toxic variant. Here we're looking specifically at a gene expression of kidney injury molecule 1. So this is a prominent kidney injury marker. And you can see here it goes up with just microcystin treatment, but significantly decreases when you follow that up with the probiotic treatment. And here we're looking at liver injury. So again, OSMR, a potent liver injury marker. You can see increases following microcystin treatment, and then significant decreases following treatment with the probiotic. And then CD36, which is a marker of lipotoxicity or fat accumulation in the liver. You can see again increases with microcystin and then significant decrease with probiotics. So here I just want to talk briefly. So this is a paper we recently published in the University of Toledo's Journal of Translation. Probiotic pretreatment protects kidneys exposed to microcystin LR. So just to give you a big picture overview of the study, so we had a control group with water, then followed with microcystin LR, then water, microcystin LR with the probiotic, and then we also had a heat inactivated probiotic as a control. We then looked at differential gene expression data from the actual kidneys and then subjected that to bioinformatics analysis. And for simplicity today, please feel free to review the paper. We are going to focus on the microcystin versus vehicle and microcystin plus probiotics group. So here what you're looking at is an overall heat map just to give you a big picture of the gene expression. And you can see here on the left, everything that's red and increased following microcystin treatment. When you add in the probiotics, those pathways or genes get significantly downregulated here in purple. So here we're looking at that suggest that key cellular signaling pathways responsible for probiotic protection against microcystin LR. And you can see here in particular electron chain transport, NADH dehydrogenase, and ribosomal subunit signaling all go up after microcystin treatment. And they all go down when you follow that up with treatment with the probiotic. And interestingly, what also goes up with the probiotic is the cytochrome P450. That's very important for detoxification. So then when you look at the bigger picture, this is a pathway analysis that suggests key signaling pathways responsible for probiotic protection, protect against MCLR toxicity. One of the top hits was the epidermal growth factor receptor. So this indicates that this could be a key target for a therapeutic approach. So with that, I know we're coming up on time here. So just to summarize, treatment of human hepatocytes with MCLR degrading bacteria isolated from Lake Erie significantly reduces genetic expression of markers of hepatotoxicity. That was the OSMR in inflammation, TGF-beta, after exposure to microcystin LR. Our in vivo studies show that pretreatment of mice exposed to the toxin with MC degrading bacteria improves the metabolism of the toxin as observed by mass spectrometric analysis and efficiently reduces genetic expression of the markers of hepatotoxicity and liver injury, namely OSMR and CD36, as well as kidney injury molecule 1. And the data we just discussed, MCLR degrading bacteria alters several signaling pathways in the kidney that can be further investigated for therapeutic targets to combat microcystin-induced kidney injury. And this data suggests that microcystin degrading bacteria could potentially be used as a therapeutic to attenuate the adverse effects of microcystin exposure. So with that, I'd like to show, here's a picture of our group. We were fortunate enough to work with a lot of outstanding medical students, undergraduates, medical residents. And I want to call out Apoorva Lad again in the center here, who's been outstanding and really drove a lot of these projects. And thank you to all of our sponsors and all of our support, in particular, the Ohio Department of Higher Education and the NOAA Sea Grant Program for sponsoring a lot of this work. Thank you.
All right, well, thank you. This was really a very interesting presentation, so thank you. Okay, so we had quite a few questions from people about how concerned are you with airborne algal toxins?
Yeah, I think it's definitely a concern. So we actually and I think there's even some folks on the call that we're fortunate enough to work with. So not only has the Ohio Department of Higher Education funded some studies, but also that's led into NIH and NSF funding for studies that we're involved in to answer that question. I mean, I think I don't think anybody thinks it's a good idea to be breathing them, but to address it in sort of a rigorous hypothesis-driven manner. That's what we're doing right now. And we so what we can say from the data that we have so far is that what we do is we model this using a 3D culture of human airway cells. We take biopsies from patients, and they could either be healthy, that, have donated a specimen, or who have a different, have a disease condition like COPD or asthma. And so we get those cells, we grow them up, and we grow them up not in a way that, normal cell culture is done, but since these are, lung cells, they live at what's called an air-liquid interface, right? So your lung cells aren't, constantly submerged underwater. So we grow them up in that same interface so that they can be exposed to air on one side, and then the media that supports them on the other. And then we have an aerosolization system where we can aerosolize different toxins over top of those and see how they respond. So from those studies, which again are, they're as close as we can get to simulating what would happen, we see that it, even in healthy cells, you won't see, after they've been exposed to, say, for instance, microcystin, you won't see what we call phenotypic or observable with your eye changes in how those cells behave after very short-term exposure. So this is 3 minutes a day for 3 days. So these cells have cilia, and you look at how they beat, or they interact with each other. So you look at how closely those cells link together, and that appears to be intact after those short-term exposures. But in those same cells, if you look kind of underneath at what's going on, you see a lot of inflammatory pathways being upregulated. And then if you take the media from those cells and you look at things that those cells spit out after they've been exposed to aerosols, and you say, what's the impact on other cells? So for instance, in the lung, when you get an irritant, you will have immune cells that will naturally want to come in to handle that. And that's done by the lung cells sending out like a danger signal. And so we looked at the ability of these cells that have been exposed that appear normal, but do have an inflammatory profile, to interact with other cells like neutrophils. Those are some of the immune cells. And what we see is that there's actually a really potent attraction of those neutrophils to the lung cells. So we think that, at least conceptually, in our system that's as close as we can get to what's going on out in the real world, that there are some suggestions that this, even in a healthy setting, that this is bad. And then if you take those same cells, but you isolate them from patients with asthma, we're seeing preliminary indications that exacerbates. So not only, in the healthy cells, you don't necessarily see the cilia beating at a different frequency. You don't see mucus production that is, above normal. But in the cells from asthmatics, you definitely do see those phenotypic changes, the cilia beating differently, the mucus production being increased, and the overall inflammatory profile increase. So that was a pretty long-winded answer, but that's what we know. And then the second part of that is kind of stay tuned. We're really grateful to state of Ohio and to the NIH and NSF for funding studies where we can, we'll be doing and I should put a plug in, because our IRB just approved it, a community health study in the area where we, invite people from the counties in Northwest Ohio to participate in a long-term community health study to look at how they, feel and how their lungs function and perform before harmful algal bloom season, during harmful algal bloom season, and then after harmful algal bloom season. So that we're really, really grateful for that support, because we think it's an important question that needs to be addressed in a really rigorous manner.
And I'll make sure to add the harmful algal bloom research initiative annual report into everyone's emails, and it has a nice summary of several of these researchers' projects, including the ones that Dr. Kennedy had mentioned that deal specifically with aerial, why do I call aerial? Airborne, sorry. I don't know where that came from. Airborne toxin. So I will make sure to send that to everyone this past annual report. Another question that we had was dealing with the microcystin degrading bacteria. And this is pretty specific, so let me just literally read it. So do microcystin degrading bacteria specifically degrade microcystin or degrade a wide range of cyclic peptides, and what, or I guess cyclic peptides, and what components of microcystin are used to sustain their growth?
Yeah, that's an excellent question. So what I can say, so this is these bacteria, we have to give full credit for the discovery and the studies behind them to researchers at the University of Toledo, and then also I think Kent State University that participated in this. And so doctors Huntley and So and King, and what so I'm going to be putting words in their mouth, but I do it all the time anyway, so we'll if I say anything wrong, hopefully they'll correct us. But these bacteria have been selected for by their ability to degrade microcystin. So essentially what happens in the experiments that where they were isolated is they were forced to only have microcystin as a food source. So there was nothing else. So they took all the bacteria that were present in the lake, and then essentially it's a natural selection where the bacteria that can't eat microcystin die, and those bacteria that could were enriched. And so again, in Dr. Huntley's lab, they isolated about 5 different bacteria. Some of those were, I should say that the characterization of that is ongoing. So they're doing sequencing. They've published some data, but I think, in terms of the data sets that are annotated, they don't know exactly a specific identification on all 5 of those. And then Dr. So they have characterized the 2 bacteria. So we know that they can specifically degrade microcystin. We don't know, to be specific, if there are other cyclic peptides that they can degrade. So whether those studies are ongoing or not, I imagine they are, but we're not directly involved in that. But it wouldn't surprise me. But we do know again that they certainly are very efficient at eating microcystin and using that as a carbon source. So.
All right. Another question we got was, I guess, what would be the next steps to your research?
Sure. So we are really excited, and again, thanks to the Ohio Department of Higher Education HABRI program. We have 3 very exciting studies that we're working on right now that were recently funded. One is to look at an aging and young model following exposure to aerosolized microcystin and aerosolized microplastics. Because when you think about these toxins, they don't exist separately out in the lake. They're often found together. So that will tell us, what are the effects on an aging community versus a younger community. Another one that gets, again, back to the aerosol issue that we're really excited about. We're going to have these big air samplers. I should say a air sampler. We're going to put stationary out at the Lake Erie Center. So we'll be able to collect in all the toxins coming off of the lake, and then working with Dr. Dragan Isalovich, who's an outstanding chemist here at Toledo. He will help us characterize those toxins, and then we can move them back into the lab and look at the human health effects. we have a very special system that specifically looks at aerosolized toxins. So we're really excited to do that, and we're using a number of different human cell lines to look at those health effects. And then finally, we have one that's going to dig down deeper into the into the diagnostic, or creating help develop tests for microcystin exposure, not just in humans, but also in animals as well. So for veterinary applications, and that's kind of a long-winded answer, but those are our next steps, and we're really excited and feel very fortunate to be able to do this work.
All right. Well, I think we have the majority, if not all, of the questions answered. I don't see any more in the chat here. So I think we will go ahead and wrap up. I wanted to, again, thank Dr. Kennedy, Dr. Holler for their willingness to talk with us about their harmful algal bloom research. This was really an excellent presentation, so we really appreciate your time. I also wanted to remind everyone that our survey URL is in the, for this webinar is in the chat feature, so please take a few minutes and fill that out. This webinar series is sponsored by Ohio Sea Grant, and will continue next month with Dr. Chris Winslow from Ohio Sea Grant and Stone Laboratory, who will be giving us a HABs primer. So all the information you need to know to get ready for the HABs forecast, that is June 27th. So the registration link for Dr. Winslow's presentation is in the chat. Again, thank you, Dr. Kennedy, Dr. Holler. I always am so happy when you can get on and present your research. So this was really a treat. Thank you everyone for participating in this webinar. We hope this was beneficial and hope you'll join us again in an upcoming webinar. Thank you again and have a great afternoon. Thanks again, Dr. Holler, Dr. Kennedy.
Thank you so
Thank
much,
you, everyone.
Jill. And yeah, if you liked it, it's Kennedy, Kennedy, and that survey. If you didn't, it's Haller, Haller.
There you
Really
go.
appreciate it.
There
Thank
you
you for
go.
everybody's time.
Thank you.
Take
That's
care.
it. All right. Well, thank you again. We really appreciate your time.
Thank you.
Have a good
Bye.
afternoon.
Bye-bye.